Okay, so let's continue. So now we're talking about a more reliable method. So after you've gone through gel electrophoresis, you're letting it run, you've shown them the results, or you may go on to this before you get to the results of the gel while it's running. And so it says that in crime scenes we're getting very little pieces of DNA, and so sometimes it's not reliable enough to get a whole genome. But if we really want to know if they're related, like for a paternity test or something, then you can actually get a cheek swab and get a whole genome. And when you have a whole genome, there are some more things you can do besides STRs. So one of the things we can do is a restriction digest. So I would have people read the little introduction out loud. And we're talking about restriction enzymes. They cut palindromic sequences. You might be thinking, what in the world is that? So ECOR1, which came from E. coli, and it was the first restriction enzyme they found, so ECOR1, it cuts the sequence, let me change my marker, GAATTC. And you might be thinking, how is that a palindrome? Well, if you read it from the other direction, on the opposite strand, it is GAATTC. Okay, so if you have a DNA sequence, a piece of DNA, and it has that sequence in it, the GAATTC, then ECOR1 is going to target that and cut that. And it cuts it in sort of a diagonal fashion. So it cuts it right after the G, going in both directions. So what you end up with is a cut like this. And so your fragments end up being a piece of DNA that looks like this, and a piece of DNA that looks like this, with your overhanging AATT. Okay, so you can see that those would be what we call sticky ends, right? And so there's a couple of things we can do with this. Number one, we can have sticky ends so we can stick things together, so different pieces of DNA that we want to adhere together. The other thing is, if you took my DNA and threw it into a bucket with ECOR1, and you took your DNA and threw it into a bucket with ECOR1, do you think that I have GAATTC in different spots, perhaps, or more spots than you do in your DNA? And the answer is yes. So if you were to chop all of my DNA up into pieces and chop all of your DNA up into pieces, our pieces might be of different lengths and sizes. And so you can take the restriction fragments and you can run them on a gel. So if you were to run a gel, gosh, all my markers are kind of crap. Okay. If you were to run me on one and you on the other, what you might see is that I have these pieces. Wow, all of these are terrible. And you have, your pieces look like that, whereas my pieces look like this. These would all be the same length. Okay? So you can see that my fragments look different than your fragments. And we call these restriction fragments. Length polymorphism, meaning the lengths are different between us. And so we abbreviate this to RIFLIPs. So my RIFLIPs are different from your RIFLIPs, and that's a DNA fingerprint. All right, so that's one thing we can do with it. So if you explain that and then you say, okay, go ahead and do 5 and 6, right? They should be able to, on question 5, they should be able to cut that sequence. They just have to find the GAATTC and then cut right after the G. They can either count the fragment size on the top strand or the bottom strand. It doesn't matter. They'll be off by just a few letters. So they count the top or count the bottom. And then number 6 asks them, how might these restriction fragment lengths be beneficial in identifying people? And I've just explained that to you. So you might actually wait and do this explanation until after they've had a chance to think about it in number 6. Then you can draw out this DNA fingerprint. All right? Okay, so now comes the tricky part, and this is the insulin application. So you can say, now that we know how to manipulate DNA, we can cut it apart, we can sequence it, we can run it on a gel and figure out its size, how can we use this to make insulin for diabetic patients? Okay, so they're going to be using plasmids. And you'll want to explain, and it's in their introductory information, that plasmids are extragenomic pieces of DNA, meaning that they're not part of the original bacterial chromosome. So if you look at a bacteria, if you have a bacterial cell, they will have their chromosome, right? And then they have these little tiny extra ones that are called plasmids. Those extra chromosomes, That's what we're dealing with. They're little plasmids, okay? And so plasmids can be used. We can splice plasmids open and we can put pieces of DNA in them of whatever we want, and then we can put them into bacteria. Well, when bacteria do mitosis, they'll copy their gene, but they'll also copy all the plasmids. So you can see the more bacteria we grow, the more plasmids we can get. So that's what we're going to do with this insulin gene. All right, and so number 7 asks, how might plasmids be used to produce human insulin? What assumptions are we making? So let students sort of think about this. What would they actually do to create human insulin in a bacterial cell? And the major assumption that we're making is that bacteria will read DNA and deal with DNA the same way that we do, which turns out they do, so it works. So down here, there's a picture of the plasmid. We're going to use a very specific plasmid. It's called the PGEM-T-Easy. It's an Invitrogen product, and it's used specifically for PCR products because the PCR enzyme, Taq polymerase, when it creates these pieces of DNA, it actually tacks on an overhanging adenine base. It's just a funky thing about this particular Taq polymerase, or Taq polymerase in general. And so we can use that to our advantage. So Invitrogen, this company, has made an artificial plasmid that has overhanging T's on it, which is why it's called the PGEM-T-Easy, okay, so that we can easily ligate a piece of DNA into it, all right? So you're just going to sort of explain that. A couple other things about the plasmid. It has transcription sites like TATA boxes. So TATA box is what we use for RNA polymerase. Bacteria use different sequences, so there's a sequence here and there's a sequence here for 2 different DNA polymerases. There's a T7 and there's an SP6. That's the equivalence of our DNA polymerases. They just have several different ones with different TATA boxes, different promoters. Okay, so the promoters are on there. There's a couple other things about it we'll talk about. So number 8 asks them to consider all these things. Okay, so I'm just going to go through each of those. You're obviously going to let them consider them on their own, maybe each one individually, and then talk about it. But I'll go through them very quickly. So A, it says, how would you amplify just the insulin gene and not the entire genome? So we're going to make our insulin gene. This is our insulin gene. We're going to sample a human, get their whole genome, and then we want just the insulin gene. So how would you do that? We'll remind them about PCR. So we're going to amplify it using PCR. How do we target just insulin? Well, that's going to be your gene-specific primers. So you're going to make a primer that sits down right here and a primer that sits down up here so that you copy just the insulin gene. Okay? So that answers part A. Part B says, what enzyme from DNA replication might be useful in getting your PCR product fused into a bacterial plasmid? They may not be able to answer this one because we're not doing DNA replication in the lab. Okay, so the answer to that is ligase. So ligase is what glues the Okazaki fragments together in DNA replication. And so you're just going to have to tell them. There's an enzyme, you'll learn about it in class, that glues together the DNA fragments. It's called ligase, okay, because it ligates DNA. For C, it says, once you've created your transformed plasmid, how will you insert it into a bacterial cell? So at this point, you want to review the steps that they've probably already done. So researchers would throw in the whole human genome along with gene-specific primers and run PCR, so that you end up a whole bunch of these. Run it on a gel to make sure it's the right size, that you really think you've amplified it. You can cut it out of the gel, get that DNA. Okay, now we want to throw it into a plasmid. So that process is called a ligation. So we take our plasmids that we've purchased from Invitrogen, and we take our PCR product that we've made, and we throw it into a test tube along with ligase, and we just incubate it at body temperature for a little while, and it will glue this into this. Okay, so what you'll end up with is a plasmid with the insulin gene stuck in it. Okay, so that is a ligation. Then you're going to put that into a bacteria, and that is called transformation. And so there's a couple ways that we can put it into a bacteria. One of the ways that we commonly use is to stress the bacteria out, because when bacteria are stressed, they tend to suck in external DNA trying to find a solution to the problem they've found themselves in. And so we actually, to stress them out, we'll put them in ice, and then we'll put them in heat, and then we'll put them in ice, and then we'll put them in heat, and they freak out and suck in DNA. Okay? The other way you can do it is you can actually use like electrophoresis. Because DNA is negatively charged, we can actually shock it into them. So you take the bacteria and stick it between 2 electrodes and send a bolt of electricity, and it will send these little plasmids like bullets shooting into the bacteria. Problem with that one is that sometimes it shoots right back up the other side of the bacteria. So there's two ways of doing it, but once you get this plasmid inside a bacteria, C asks, once you created it, how will you insert, oh, that's C, that's how you'll insert it into a bacteria. In D, it says, how will you select for bacteria containing your plasmid? So you've got to think about it. You've got this tube of bacteria, thousands and thousands and thousands of cells. You drop in some of these, only some of them are going to get these, and you only want to grow the bacteria that have these. So that's where our ampicillin resistance gene comes in. So on the plasmid, and they may not have noticed this, but when they look back, there's a little AMP-R gene, that's for ampicillin resistance. These bacteria, we've given them the resistance to the antibiotic ampicillin. That way, when you're growing bacteria, all you do is dump a bunch of ampicillin into the culture, and any of the bacteria that didn't get a plasmid, they're not going to survive. So you're only growing ones that have a plasmid with your gene of interest in it. Okay, so that's D. And then E says, how will the bacteria produce human insulin? We're making the assumption that bacteria will transcribe that the same way we will and translate it the same way we will. And they do. Now, so what they're going to do is they're just going to translate it. And then we'll just isolate that product, the protein product, from the bacteria. But if we were going to transcribe it in a test tube, so let's say we have some concerns that the bacteria maybe don't read it exactly the way we do, we can isolate this plasmid, and we can use SP6 or T7, and we can make insulin ourselves in a test tube, okay, as long as there's amino acids present. And so in that case, you want to know which orientation it's in. And there's a really ingenious way of figuring out orientation that seems to really trip students up. But this is where they're going to use their plasmids from their packet. They're in the packet. I don't know if Clarice is going to cut out some for you, the lab tech, but here's your plasmid. So I've got a mock plasmid here. So this is one part of it that's going from the 3600 side, and then this is the other side that's going 1 to 1800. Okay, so if you take this and you tape these together, all right, this would be your PGEM TEZ plasmid. Okay, so it would look like that circle that's on the board going around like this. All right, so this is your PGEM TEZ. I'm going to make 2 of them. All right, so I'm just taping it together so it goes 1 to 3600. All right, okay, so I've got my 2 plasmids. And the reason I'm making 2 of them is that I tell students, okay, so pretend you have this plasmid, just like it is on the board, and I've got my insulin product. It's also in their packet, okay? Got my insulin product, tape it in as if you're doing the ligation. Okay, so in this one, if you notice, I'm going to tape it so that this is 1 of the plasmid, and this is the gene right side up. So the ATG, the start codon, is right next to the 1. So I'm going to tape this one like that. I'm going to tape it around in a circle. Okay, but it's a 50-50 probability of which way it will go in. So this one's gone in so that the ATG is next to the 1. Okay, but you could also just as easily have taped it in with the ATG next to the 3600 side. So like that. Okay, so this one I'm going to tape in the other way. So we have 2 different orientations. If I was going to make this in a test tube, I need to know for this one, I want it to read it this way, so I'm going to use the T7, whereas for the other one that's upside down, I got to use the SP6 to read it, okay? And so otherwise you're going to make some weird protein that's the template strand. You're going to make the wrong mRNA. All right, so here we have these 2, and they're equally probable. So how do you figure out which orientation yours is in? Well, you simply harvest a plasmid, and then you throw in a restriction enzyme. So in this case, we're going to use SAC2, because you can see that the plasmid has a SAC2 site. It comes from Saccharomyces. Okay, I need to grab a pair of scissors. One moment. Okay, so if I were to throw in SAC2, it's going to cut right at that base pair 40 in the plasmid. Obviously this is not to scale, this is 40 letters long. So it's going to cut right there. Okay, but there also happens to be a SAC2 site in the DNA. It's a CCG-CGG. So it's about right here. Okay. So if I threw SAC2 in with this orientation, I'd get these two fragments. If I threw it in the other orientation, it's still going to cut at that CCG-CGG site in the gene. But in the plasmid, it's going to cut on the other end over here. So I'm going to get these two fragments with this orientation. So if I were to run them on a gel, okay, so let's say this whole board, well, we'll do it in a second. Here. Yeah. If I were to run them on a gel, it's a giant gel on my board. And I run one orientation, I run the other orientation. Where would it end up? So here was our first one that we did. Okay, so this little tiny piece, is it going to end up at the top or at the bottom? Well, because it's small, it's going to run all the way to the bottom, right? It's going to run really fast. So this one would end up somewhere here at the bottom, and then this giant piece is going to end up somewhere here at the top. Okay, so it'll end up at the top. So then you have to ask the other orientation. Here's my two fragments. So this fragment, it's also short. Where is it going to end up? Well, it's not quite as short as that one, so it's going to end up a little bit higher than this other piece, right? So it's going to end up a little higher. Whoa, that was kind of crooked. And then this one is a little bit shorter than that one, so it's going to end up a little bit lower. So if you ran the two on a gel side by side, you could differentiate between which orientation you've got, whether it's higher or lower. Now, could you tell me exactly what size these fragments are? Well, of course you can. So it turns out, so this orientation, this one had the ATG next to number 1. So if we drew the plasmid, can you see this? Yeah, okay. This is 1, this is 3600. Our gene is in there, the insulin gene, and in this case the ATG is on that side going down. Okay, so when we cut this one, we cut it at right here, and I think if I remember right, that's at about base pair 19. And I can't remember how many's after that, but let's just do round numbers just for kicks. Let's say it cut at base pair 20, and that there's 60 over here. Okay. Now, it also cuts at 40 in the plasmid, so there's 40 here, and then how much is the rest of it? Well, it's 3600 total, so it's 3560 is the rest of it. So, this fragment, this little fragment right here, how big is it? Well, it's these 40 plus these 20, so it's 60. And then how big is this other fragment? Well, it's 60 plus the 3560, so it's 3620. Now, keep in mind, these are not the right numbers. I haven't counted the gene to know what it is. Okay, so this one is up here around 3620. Then we could do the same thing with the other orientation. So in this case, the orientation is upside down. So 1 to 3600. And this gene, in this case, is going with the ATG in this direction, okay? So in this one, we're cutting it again at the 40. So this is 40, and this is whatever was left, 60. I'm sorry, 20, my bad. 20 and 60. And then it's again cutting at the 40 here, so this is 40 and this is 3560. So how big is this fragment? Well, it's bigger, it's 100. So this one is a 100 base pair fragment, and this one is 3560 plus 20, so 3580. Okay, so you can figure out the exact. Now I think in this particular gene, the cut site in the gene is like at 19, and this is maybe 47, but I can't remember. You'll have to go look. All right, so that's it. I know that's kind of confusing, but try to work with the students on it. You should have plenty of time to do that, especially with not doing DNA replication. Make sure you have them look at the gel. The gel should work beautifully. I'll show you. You'll have a picture of it. And we also put the template in the refrigerator if you want to look at that. Okay, let me know if you have questions.